link for my previous video on biomolecules. We are doing Unit 14 from Class 12, Chemistry, Biomolecules. And the first part I have already uploaded. And today in this video, we are going to continue with Unit 14, Biomolecules, CBSE Class 12. Please do like and subscribe to my channel and don't miss out my introduction video. Those who have not watched, the link is given in the description box. So first, we will have a quick revision of proteins. What are proteins? Proteins are actually polymeric chain of amino acids. And what is protein composed of? They are made of long chain of amino acids. So here in this screen, we can see the important functions of proteins. Proteins serve many vital functions in our body, and they actually give our body structural support, and they help in speeding up of chemical reactions, and they also help in transport of molecules and many more. Here we'll have a quick glance about some of the important functions of proteins. And proteins, firstly, they were known as polypeptides, and it is made up of elements like carbon, hydrogen, oxygen, nitrogen and sulfur. And proteins are made up of many amino acids bonded together in a long chain, and they have structural and mechanical uses, and they participate in many cellular processes. Their main function is as enzymes, also as pigments and steroid hormones. And now we'll learn about the importance of essential and non-essential amino acids. First of all, what do you understand by essential and non-essential amino acids? Essential amino acids are amino acids which cannot be synthesized and which must be obtained through diet. And non-essential amino acids are those which can be synthesized, or the body can make it inside the body itself. And amino acids are classified as acidic, basic or neutral based on the relative number of amino and carboxyl groups in them. And here you can see a list of essential amino acids and non-essential amino acids. Essential amino acids includes arginine, histidine, isoleucine, leucine, lysine, methionine, phenylalanine, threonine, tryptophan and valine. The non-essential amino acid includes alanine, asparagine, aspartate, glutamate, glycine, serine and triosine. And you should know that the structure of amino acid is extremely very important, and many questions of NEET are based on the structure and uses of amino acids. So first of all, as you can see in the figure of this structure, amino acid contains amino, that is NH2 group, and carboxyl COOH functional groups. Depending on relative position of amino group with respect to carboxyl group, amino acids can be classified as alpha, beta, gamma, delta and so on. Only alpha amino acids are obtained when you hydrolyze proteins. All alpha amino acids have names that reflect the source of property of that compound. For example, if you take glycine, it is so named as it has a sweet taste. And now again coming back to proteins, proteins are polymers of alpha amino acids, and they are connected to each other by peptide bonds. And what is peptide bond? Peptide bond is actually an amide formed between carboxyl and amino group. And you can refer to the structure in the text as well. And here you can see the structure of protein. Amino acids are bound together through dehydration synthesis. And the carbon group of one amino acid that you can see on the left hand side, it binds to the nitrogen group of the second or another amino acid that is shown in the second figure. So these linkage or these bonds are called peptide bonds or peptide linkage. And a reaction between two molecules of similar or different amino acids proceeds through combination of amino group of one molecule with carboxyl group of other. So as a result, water molecule is eliminated and peptide bond is formed. And in this figure, you can see a polypeptide with more than hundreds of amino acids. It combines with molecular, which has got molecular mass higher than 10,000 unit is called protein. So now again coming to sweeter ions. What are sweeter ions? In sweeter ion form, amino acid shows amphoteric behavior as they react with both acid and bases. So here you can see the structure of sweeter ion. So due to internal transfer of a proton, what is a proton? It is a hydrogen atom from the carboxyl group to the amino group. Amino acid has, it shows both negative charge at the carboxylate group, and you can see in the figure here, and a positive charge at the ammonium group. So it just resembles a salt like ion. That means it has got positive and negative both charges together, and so that is why the salt like ion is called a sweeter ion. So amino acids are actually crystalline solids and they have high melting points. Why they have high melting points? Because they are having some ionic character. So due to the salt like nature. So in solutions, the charge of the amino acids changes as you can see in the figure. So in different solutions, the charges are different. So except glycine, note that all other naturally occurring amino acids are optically active. Why? Because here the alpha carbon atoms is asymmetric. So there exist both D and L forms. So I hope you understand about sweeter ions and their importance, their structure, and we learnt about why they have a sort like ion structure because of the presence of both negative as well as positive charge. And next we move on to the classification of proteins which are done on the basis of their molecular shape. So here you can see there are two forms. One is fibrous and the second one is globular. What is fibrous protein? When polypeptide chain runs parallel and if they are held together by hydrogen and disulfide bonds, then fiber-like structure is formed and these are insoluble in water. For example, keratin, which is present in air, wool, silk, etc. And then now globular protein, here chains of polypeptide coil around to give a spherical shape and it is soluble in water. So here in this chart you can see the different shape, purpose, sequence, durability, example, solubility, everything, and as a comparison between fibrous and globular structures. And you can see fibrous, the shape is long and narrow, whereas in globular it is round and or spherical. Then the purpose of fibrous is structural, globular is functional. In acid sequence in fibrous, it is repetitive amino acid. It repeats, amino acid sequence gets repeated. Whereas in globular structure, irregular amino acid sequence can be seen. And durability in fibrous, it is less sensitive to changes in pH, temperature, etc. But in globular, it is more sensitive to changes in pH, temperature, and all that. Then examples of fibrous structure is collagen, myosin, fibrin, actin, keratin, elastin, etc. And globular examples you can see enzymes, hemoglobin, insulin, all these names you might have heard of and learnt in biology, and immunoglobulin. Then finally, regarding the solubility, fibrous, as we already said, it's generally insoluble in water, whereas globular form is generally soluble in water. So you just read this table thoroughly, and you have to be familiar with all the terms and all the structures, everything, because questions can come anywhere from this topic. And next, what are the four phases of proteins? can see that proteins have four levels of structure, which are primary, secondary, tertiary, and quaternary. And in primary, you can see that the amino acids are bound together and it has one or more polypeptide chains. Whereas in secondary refers to shape in which a long polypeptide chain can exist and shows two different structures called alpha helix and beta pleated sheet structure. And here individual amino acids are bent and molded as needed. Then comes the tertiary structure, which represents the overall folding of polypeptide chains, which gives rise to two major molecular shapes, and that is fibrous globular. So here the entire chain of amino acids is bent and molded as needed, forming a subunit. And finally there is quaternary structure, where some proteins are made up of two or more polypeptide chains called subunits. So multiple completed subunits are fitted together to make a complete protein. So we have already have had a quick revision on the four phases of production of proteins, and moving on to the structure which we explained before. You can see that in this figure it is very it very clearly explains the different structure. Like primary structure, you can see amino acid. Secondary structure, that is helix. What happens in the secondary structure, you can see the shape like long polypeptide chain. It bends, like you can see that. And then the next tertiary structure, in which overall folding is shown. And finally in quaternary structure, two or more chains, you can see they are forming the subunits. So you can go through this figure and revise it well. And next topic, we are moving on to denaturation. And what is denaturation? It's a very important process. uh, Here we can see that a protein loses its shape, native shape, because the weak chemical bonds which are present are disrupted, and they become biologically inactive. So what happens in the case of proteins is that the three-dimensional structure is lost, and it is sufficient to cause the loss of function as well. And secondary, tertiary, quaternary structure of proteins as well are lost. And then the change in physical, chemical, and biological property of protein molecules all are affected. And what is the definition of denaturation? It involves transformation of a well-defined folded structure of proteins formed under physiological conditions to an unfolded state under non-psychological conditions. That is called protein denaturation. Initially the shape of the protein, it is well defined and well folded, and it was formed under psychological conditions. But now after denaturation, what happens? That is, its native shape is lost, and due to the breaking of the chemical bond, and now it becomes, as you can see on the right hand side in the figure, it becomes unfolded, and then the full change or transformation occurs. So that is an important process of proteins. And next, we'll move on to a very important topic called enzymes. All these topics you might have already learned in biology as well. What I mean to do is, not to teach the unit 14 biomolecules of class 12, but this is done as a quick revision, so that you can really find the MCQs coming from this lesson easy to solve, and you can score good marks by just revising all these topics. So that is why I'm not going deep into the subject, just only an overall revision I am doing. So now we'll move on to enzymes. So what are enzymes? Enzymes, you have heard that word often, that is, life is possible because of the coordination of various chemical reactions in living beings. For example, you eat food, and after that what happens? Digestion of food happens. Then the absorption of nutrients and suitable molecules of food takes place, and then finally what does it result in? Energy production. So you see that all these reactions occur in mild conditions in our body with the help of some biocatalysts, and these biocatalysts are called enzymes. Actually, what are they are globular proteins, and enzyme that catalyzes hydrolysis of maltose into glucose is called maltase. So you can notice here that the name of the enzyme normally ends in ASE. And you can see in this figure, they speed up the digestion and metabolism. So where can you see enzymes? Normally they are located in the cells, cytoplasm, mitochondria, tissues, body, fluids, etc. Then endoenzymes and enzymes which function within the cells. So what are endoenzymes? There are two types of enzymes. One is endoenzymes and one is exoenzymes. So endo means inside, within the cells function happens. So you can see that in our body most of the enzymes are these types. For example, metabolic enzymes like cytochrome oxidase and all that belongs to this category. Exoenzymes, external, outside. So these enzymes are liberated by the cells, and what they do is that they catalyze the reactions outside the cell. For example, digestive enzymes like amylase, lipase, protease, etc. belong to this category. Now we have learned about what are enzymes, what is their importance, what are their functions, where are they located, and what are their classifications and examples as well. So I hope you understood this. Now we'll see what is maltase. So you can see that the small intestine walls, they have got certain glands which produce enzymes known as maltase. So you can see that maltase will break down maltose into two glucose molecules. You can see that in the figure that you can see in maltose structure. Then maltase above the arrow you can see, which is the enzyme, helps in breaking down maltose into two glucose molecules. So you have to thoroughly revise the entire NCERT lessons because you get questions from each and every line for NEET examination, and mostly you won't get direct questions, so you have to be thorough with the concept. So read the textbook thoroughly, and then you'll find the questions really easy and simple. So now we'll move on to vitamins. So what are vitamins? So certain organic compounds are needed in small amounts in our diet, but their deficiency causes specific diseases. So these compounds are called vitamins. So mostly the body cannot make vitamins which are needed for its function, but bacteria of the gut can produce certain molecules, vitamins. And now we'll move on to the definition of vitamin. That is an organic compound required as a nutrient in tiny amounts by an organism. Then we already said that it cannot be synthesized in sufficient quantities by an organism, and it should be obtained from the diet. So vitamins have diverse, so many functions, biological function. So a few I've mentioned here. First one being hormone-like functions, like they act as regulators of mineral metabolism, like vitamin D and all that. Then they are the regulators of cell and tissue growth and differentiation, like some forms of vitamin A. Then antioxidants like vitamin E and C. Then enzyme cofactors, like tightly bound to enzyme as a part of prosthetic group, coenzymes. They are also known as coenzymes. So here vitamins are divided into two. One is fat soluble vitamins and water soluble vitamins. So here you can see in this chart the classification of vitamins: fat soluble vitamins and water soluble vitamins. So fat soluble vitamins are vitamins which are soluble in fat and oil but insoluble in water. And water soluble vitamins, they must be supplied regularly in diet because they are readily excreted in urine and cannot be stored in our body. Exception is B12. So you have to remember the exceptions as well, because you'll get such questions for NEET exams for sure. And what are fat soluble vitamins, it includes vitamin A, vitamin D, vitamin E, vitamin K. Then water soluble vitamins, they include vitamin C, thiamine, riboflavin, niacin, pantothenic acid, then pyridoxine, folic acid, vitamin B12, biotin, etc. So for the exam, they'll be asking the name of the vitamin and which category it belongs to. So you have to be thorough with this table. And this is one of the most important topics of biomolecules, which is highly, asked for the NEET different years. You have to learn this table thoroughly, and you can see that the name of vitamins and the deficiency diseases caused by each of them. First one is vitamin A, and the deficiency of vitamin A or the lack of the vitamin A causes xerophthalmia or night blindness. And vitamin B1 deficiency causes beriberi. And they'll ask you the deficiency of which vitamin causes beriberi. You have to know that it is vitamin B1. Then vitamin B2, it's also known as riboflavin. Then deficiency diseases caused are chelosis, digestive disorders, burning sensation of the skin, etc. Then vitamin B6, that is pyridoxine. Pyridoxine causes convulsions. Then vitamin B12, pernicious anemia. Vitamin C, also known as ascorbic acid, causes scurvy. Vitamin D causes rickets and osteomalacia. So you have to learn the names of all these diseases, the name of the vitamins, etc. thoroughly. Now, next topic is nucleic acids. So what are nucleic acids? So nucleic acids, like each species, even if it is human being or whatever species, resembles our ancestors in many ways. So the nucleus of a living cell is actually responsible for transmission of inherent characters, which is known as heredity that you have learned in biology in detail. So the particles in nucleus of cell are responsible for heredity. They are called chromosomes, and they are made up of proteins and nucleic acids. So there are two types of nucleic acid. One is deoxyribonucleic acid, DNA, and ribonucleic acid, RNA. You can see the structure given here, and you can just notice the numbering of carbon atom given for each structure. From the right hand side, middle starts 1, then down is 2, left side bottom is 3, then you have got 4 and 5 position. So you have to remember the structure thoroughly. And next we will see that here a unit formed by attachment of base to position 1 of sugar is called nucleoside, and when nucleoside is linked to phosphoric acid at 5th position, it forms nucleotide. So this is very important. When nucleoside is linked to phosphoric acid at 5th position of sugar molecule, we get nucleotide. So actually you have to learn the difference between nucleoside and nucleotide. So next you can see the structure of the nucleotide. You can see actually it has got a phosphate group, a sugar and base. So 3 categories add up and finally you get nucleotide. And actually James Watson and Francis Crick, they were the 2 scientists who gave double-stranded structure for DNA, which is very important. And 2 nucleic acid strands, which are complementary to each other, are wound together by hydrogen bonds between pair of bases. Here you can see base shown in the figure, and then you can see position 1 to 5. And in the second figure, you can see the phosphoric acid linkage to the 5th position of the molecule. So this structure is also very important, and you have to know that well. And finally here in this figure you can see the DNA structure, and you can see that is for your simple and easy understanding. I have given a colored chart. So you can see from this figure itself that adenine can combine only with thymine. Look at the colors of the figure, and then purple and magenta, that is cytosine and guanine. So green and magenta, adenine and thymine. And you can see that brown color, phosphate, is the base or the backbone. And these strands are wound and held together by hydrogen bonds And between the pair of bases, we can see the hydrogen bond here in the figure. And the structure shown, it is known as the DNA double helix. So as I said earlier, it contains two strands wound around each other, and DNA is normally made up of nucleotides. As we already saw, it consists of a phosphate group, a pentose sugar, and a nitrogenous base. And more importantly, it contains four nitrogenous bases, or four in number actually, and these are attached to the inner sides of the strands. So the bases are attached to the inner sides. As you saw, there are four bases: adenine, thymine, cytosine, and guanine. So adenine pairs with thymine with two hydrogen bonds, and guanine pairs with cytosine. Guanine and cytosine pairing is done with three hydrogen bonds. And in DNA, a purine base always pairs with a pyrimidine. There are two types of bases, one is purine and one is pyrimidine. The number of purine bases in DNA will be always equal to the pyridine bases of DNA. And also A plus G, that is adenine plus guanine, will be equal to T plus C, thymine plus cytosine. And the nitrogenous bases of DNA are widely classified as purine and pyrimidine bases. And the pyridine bases are adenine and guanine, and pyrimidine bases are cytosine. Thymine and uracil is naturally seen in RNA. DNA doesn't have uracil. That point you have to remember, and which is lacking in DNA, and which is not there in RNA. So hope this is clear. Thymine and uracil occur only in RNA, where thymine is there in both DNA and RNA. Uracil is seen only in RNA. So now we move on to our next topic, hormones. So hormones, you might have all heard, is absolutely necessary for human body. And what are actually hormones? Medical definition is given here. It is a chemical substance that is produced in the body, which controls and regulates the activity of certain cells or organs. So many organs are secreted by special glands, like thyroid hormone is produced by thyroid gland. So many hormones are there. And finally, with that, we'll come to end of this session. Hope you found this session really useful, and I would suggest that you go through the session repeatedly. As I said in my intro video, that recorded video, why I'm not doing live sections is because recorded video always helps a student to go through the content which you are finding it difficult or confusing. You can repeatedly go through it, and whatever doubts you have, you can clear it, because I've given a simple explanation so that all of you can easily understand the whole content. Whatever content is given in the lesson in your textbook, it is clearly explained, and I have highlighted all the important points. And moreover, I'll be giving a detailed MCQ with so many questions from the previous year's NEET question papers, and very important ones. And that's all for the session, and I'll be uploading my in my next session the most important MCQs from this lesson. And thanks for watching this video.